Hello everyone, I'm Alex Ivanov, and for my AP Bio project, I will be discussing the discovery of the widely renowned genome editing tool known as CRISPR, or the longer version, which is known as the Clustered Regularly Interspaced Short Palindromic Repeat. Now, CRISPR combined with a warhead called a Cas9 protein serve jointly as a system to manually edit an organism's genome by targeting a specific section of an organism's DNA to modify the genetic code which ultimately makes up our phenotype. Uh, Now, the Cas9 protein works together with a targeting mechanism called the guide RNA, which literally serves as the guide for the Cas9 protein to find the specific section of the genome that the guide RNA is directing it to. Together, the protein and the targeting mechanism serve as a guided missile, for simpler terms, or CRISPR in scientific terms. Now, when trying to CRISPR an organism's genome, the guide RNA and Cas9 protein complex search along the organism's DNA for the section that matches the guide RNA. Then a series of events ensues to edit the genome. Firstly, the missile-like complex damages the DNA by splitting the double strands of the double helix. Then after splitting the strands, the Cas9 protein is triggered to cut the section. After that, we cause cellular processes to make the repairs we want to the DNA as opposed to a natural restoration. Those processes can be one of two major repair pathways consisting of the body's first responders. However, the first pathway which simply conjoins the two strands, but that isn't always the most effective or efficient method because it could easily result in the addition of an unnecessary base, the deletion of a base, the loss of a base. However, the second repair pathway is much more interesting because it pertains to the use of a homologous piece of DNA. Because people are diploid organisms, which simply means that we have a copy of our genome from both our mom and our dad, uh, we can deceive the body by feeding it a false piece of DNA, just as long as it has homology on both ends, but it can have completely different genetic code in the middle, and we would still be able to insert it in the DNA sequence, and this would give us the ability to take letters out, change bases, to take bases out, or to add completely new DNA to the genome. And this entire process that I'm talking about is actually derived from scientist Alexander Bolton's studies in the immune system of an ancient bacteria called Streptococcus thermophilus. And his studies displayed an irregular CRISPR locus that was first characterized by Francisco Mojico in 1993. And within Bolton's CRISPR array, he discovered some novel case genes, one of which that was particularly extensive and had nuclease activity, now known as the Cas9 protein. And the unique quality of CRISPR is that it's proven to be far more efficient and customizable as opposed to any other genome editing tool because the CRISPR-Cas9 system is already capable of cutting DNA strands without the assistance of other cleaving enzymes, uh, therefore leaving the guide RNA only needing to be designed specifically to lead the Cas9 protein to its DNA target. And an example of a monumental achievement with the use of CRISPR in modern times is actually in a recent study published by the organization PNAS, and where a group of researchers edited a gene sequence in mosquitoes to render them resistant to the parasite that causes malaria. And as far as the long-term effects of by the way, the mosquitoes actually, they proved that the resistant mosquitoes, when they mated with each other, 99% of their offspring possessed the resistance trait, and this went true for the mosquitoes who mated with normal mosquitoes as well. But as far as the long-term effects of this experiment, only this use of CRISPR just demonstrates how fundamental this use of CRISPR can be in the prevention and the transmission of diseases between mosquitoes and insects to humans altogether. And as far as the current status of CRISPR in terms of its capability to make huge advances and change mankind is very unknown at the moment, because CRISPR is certainly going to be amazing, undoubtedly, because unlike before, with this modular targeting system, we can actually put any DNA strand exactly where we want it in an organism's genome, and we can program it the way we'd like. But while CRISPR has the ability to create what modern trends are hinting towards, or by popular demand, is the superhuman designer baby, which is achieved ultimately by genetically engineering human embryos with predetermined traits such as superintelligence or elite athleticism. However, while scientists are still exploring the realm of CRISPR to answer all the questions everyone has and the major implications of CRISPR, they fear that tinkering with the human genome like this could reap far more serious repercussions than we can currently fathom. But nonetheless, we're still working towards getting all the questions answered in order to further proceed with this new technological discovery. But that's my video on CRISPR, and I certainly hope you learned something, and I hope you have a nice day.